This video is about muscle structure and how the actual muscle works. So structurally, a whole muscle, what you would think of as a muscle if you're looking at your quadriceps or your biceps muscle, is actually a bundle of muscle cells. If we're sort of coming down inside them, we've got the bone attached to a tendon attached to that big muscle, say your biceps. You can see that this is made of lots of little bundles, and those bundles are themselves made of bundles, and what they're made of bundles of is muscle cells. So this is a muscle cell. The muscle terminology tends to begin with myo or sarco. So for example, we don't call the cell membrane this plasma membrane, it's called the sarcolemma. We've got, instead of endoplasmic reticulum, we've got sarcoplasmic reticulum. Each muscle cell, you can see, is also made of little bundles of units called myofibrils. And a myofibril is actually the functional unit of a muscle. It contains two sorts of filaments, thick filaments which are made of myosin and thin filaments which are made of actin. These myofibrils look stripy just because of the arrangement of actin and myosin inside them. So if we look at the arrangement of actin and myosin inside one of these myofibrils, myofibrils are very long and they're divided into what we call a functional unit called a sarcomere. Again, we've got that word sarco, meaning muscle. The sarcomere runs between a Z line and a Z line, and these are the structures that the actin filaments, the thin filaments, are attached to. You need to know the sort of overall structure, what goes where, and the names of all of these bands and lines. So we've got two lines in our sarcomere. We've got the Z lines at each end, and right down the centre we've got a line called the M line. The M line is where the myosin filaments are attached, these thicker filaments. The entire length of the myosin filaments are called the A band, and that's a fixed length of band. The myosin filaments don't change in length, in fact the actin filaments don't change in length either. So these filament lengths are fixed, but their relationship to each other is not. Where we only have myosin, that's called the H band, and where we've only got actin, straddling that Z line, we've got the I band. So the I band actually stretches into the next sarcomere. The overall principle of muscle contraction then is that these actin filaments are going to slide in between the myosin filaments and reduce the distance between the two Z lines. So the A band won't change length, the H band will get shorter as there becomes more overlap and less regions where there's just myosin, and the I bands are going to get shorter as more of the actin is overlapping with the myosin. If we cut sort of down and looked on these filaments end on, where we've got actin only, we would only see thin filaments, and we'd see them end on, so they look like little dots. Where we've got myosin, we would only see the thick filaments, and they would look like bigger dots. And where they overlap, we'd see a combination of both of those things. So that's the sort of structural stuff that you really have to know, and then we can look at how muscle actually works, how those filaments actually do slide in between each other. So here I've made a little model to show an actin filament and a myosin filament. So actin filaments are actually made up of three sorts of proteins. So my white pipe cleaners represent the actin. You can see that wrapped round the actin we've got a green protein. This represents a protein called tropomyosin. I know, really confusing because it's nothing to do with myosin. So this is just a straightforward fibrous protein that kind of is wrapped round. And when it's wrapped round the resting muscle. It's concealing binding sites on the actin, and these are made of yet another protein called troponin. So these are going to be the binding sites that are going to enable the myosin to get in contact with the actin and actually slide it through the myosin filaments. So the first stage in muscle contraction is actually to free up those binding sites. Just before we do that, myosin, these thicker filaments, done them in a nice thick pipe cleaner, have these little arms and these little binding bits. So they the myosin filaments actually look a little bit sort of furrier because they've got lots of projecting arms. Now these arms can move, so we refer to them as being cocked, which means that they've got ATP attached, they're in a high energy state. The ATP breaks down into ADP and phosphate, and as the ADP and phosphate leave, they will swing the arm forward in what's called the power stroke. So these are going to bind to the troponin, and as they move, they're going to move that entire actin filament along. So let me see if I can just demonstrate that for you. So we'll start with our myosin in its cocked high energy state with ATP attached. And the first thing that happens is that as a nerve impulse reaches the muscle, the sarcoplasmic reticulum, T vesicles, release calcium ions down into between all of these muscle fibres, all of these proteins. And the calcium ions bind to the tropomyosin and actually move. As they bind, they're moving the tropomyosin very slightly so that it exposes these troponin binding sites. When that happens, the myosin can then form a cross bridge. So when these are now linked together, our high energy myosin heads are now linked, cross-bridged over to the actin filaments. The ATP breaks down and these heads move, and I'm hoping that my actin's going to move with it, but it's not. I'll have to give it a bit of a push. So the actin filaments are actually moved along. ATP attaches again to re-energize these heads, and they detach. So the actual detachment is the thing that's using the ATP. They then move back and re-cock. So again, now they're in a high energy state. Bind and push. So you can see that they're just sliding those filaments. Now it's not a smooth movement because you've got to bind, push, bind, push. So it actually moves on what's called a ratchet mechanism. So it's more of a move, slide, stop, which sounds a bit like a Morris dance. Anyway, that's the end of muscle structure, and see you next time.